Our second speaker is Dr. Tyrone Hayes, professor of integrative biology at UC Berkeley. There is no greater honor in science than for colleagues to associate one's name with a discovery or more discoveries after the first one. And Dr. Hayes has accomplished that honor, demonstrating through studies on the African clawed frog and the Japanese kajika the molecular pathways through which synthetic chemicals affect gonad differentiation and steroid enzyme activities. Today, he will discuss with us his excursions with atrazine. Please join me to welcome Dr. Hayes to the podium.
Thank you. I titled this talk, my story about this scarlet letter, From Silent Spring to Silent Night, A Tale of Toads and Men, and I hope that over the next 40 minutes that the impetus behind the title will be obvious. But for those of you who don't know, as many as 70% of all amphibians globally are in decline. And my first play on words is that I think in the same way that Rachel Carson taught or believed that the death of birds and the role of pesticides were, and our pending silent spring at the time was a warning to us. In the same way I believe that our silent night, which in many places has already come, is also a warning to us. And hopefully that'll be revealed in this, again, tale of toads and men. Before I embark on that, however, as always, I'm going to do my acknowledgments first. And my first acknowledgement, as always, is to my family for their love and support. It takes money to do research, obviously. And this is an area that's been a little tricky, so I've had to go to a lot of places to get the funds, including some from the industry, by the way, who have since decided they don't really like to hang out with me, but that's okay. I want to thank all of the students that do the work. So these are students that have been with me over the last 10 years. This is a fairly recent picture of my new crew, and these guys are the guys who are currently in my laboratory and doing some of the unpublished work that I'll talk to you about. And finally, I dedicate this, as I do all my talks since 2005, to my grandmother for everything that she did and continues to do for me. So the scarlet letter, atrazine. I never heard of atrazine until the company at the time, Novartis, came to me and asked me to do some studies on atrazine. As a brief introduction, it's an S-chlorotriazine. It's an herbicide or weed killer that in this country is mostly used on corn. It's been used since 1958, and we use about 80 million pounds in the United States until recently, when it was surpassed by glyphosate. It was the #1 selling pesticide in the world. It's used in more than 80 countries, but now it's outlawed, at least in all of the European Union, or as their lawyers like for me to say, it has been denied regulatory approval. The point being that we're still using 80 million pounds of something that is now regulated across Europe. So let me introduce you, first of all, to the main character in this story. This is the African clawed frog, Xenopus laevis, and I want to take a minute to, if you've never heard of it, this is the frog that most people use in developmental biology. It's like a lab rat for people who study amphibians, for an interesting reason that many people don't know. Scientists started using this frog in the early 1900s, 1920 or so, because somebody discovered that the human pregnancy hormone would make this animal lay eggs. So until about the early 1940s, this was the pregnancy test. I tell this story for a couple of reasons. One is, it's an interesting exercise, isn't it, to wonder, who's the first guy who thought, I wonder what'll happen if I inject pee into this frog. Like, where does that come from? But it demonstrates, I believe, the importance of curiosity-based research. It led to an important discovery. And the story also illustrates how similar our hormones and our systems are to these guys. The same hormone that maintain all of the pregnancies that's responsible for everybody in this room makes this frog lay eggs. So as I tell this tale of toads and men, as I tell you what atrazine does to frogs, you should be wondering, what might atrazine do to me? And that's where we'll end up today before we depart. First off, atrazine, we found when I was working as a consultant for Novartis, that atrazine inhibited growth of the voice box of the larynx. And that was sort of bad news because male frogs sing and females don't because of testosterone. Same reason that men have deeper voices than women. And so we looked at the data in a couple of different ways, and one I think was sort of unique to endocrinologists. First of all, we showed that there was a dose effect, but it's not, as you've heard before, linear monotonic response, and I'll tell you why we believe that is in a minute. So here we're just looking at the size of the larynx as measured as it changes with the dose of atrazine. We looked at the data in a different way in our first paper, in that we also sort of looked at what proportion of animals were affected. So these data ask, how small is your larynx if you're exposed to atrazine? And these data say, I don't care how small your larynx is, how many individuals in the population are affected. So it's a non-parametric way of looking at the data. And what you'll notice is there's not a monotonic linear dose response. It's at 1 part per billion, you get an effect that doesn't decrease further with an increase in dose. That's because what we believe is happening here, and I'll show you the evidence, is that atrazine isn't shrinking the larynx, it's preventing it from growing. There's a threshold level of testosterone that's required for laryngeal growth, and once you knock out that testosterone, you simply don't see a greater effect. The larynx simply stays at its default size, if you will. So if the animals appear to be missing testosterone, of course, the next place we look were the gonads, because that's where the testosterone comes from. So here's what frog gonads look like. This is the kidney, and this is, if you've seen me talk before, you've seen this slide. This is an animal that has testes, it has ovaries, it has another testes, it has more ovaries. Whole party's going on in there. All you need. Anyway, this is, as I pointed out many times before, despite what you may have learned in Jurassic Park, frogs are not naturally hermaphroditic. So we knew there was some sort of problem with the gonads that led to both this feminization effects, because these are males that grow ovaries, which I'll talk about in a minute, and that contributed to these demasculizing effects. The proposal we made was that here's a testis, and normally a testis should make testosterone. This word literally means testicular hormone. That's where the word comes from. And our proposal was, and I want you to remember the word atrazine, our proposal was that atrazine turns on an enzyme, the machinery, if you will, aromatase, that converts testosterone into estrogen. This word literally means the generator of estrus. It is the female hormone. And as a consequence, we propose that there are two problems. One is you're demasculinized because now you're using up your testosterone. And the second problem is you're feminized because now you're making the female hormone when you shouldn't be if you're a male. So we showed, and this is from our first paper, but I'm going to show you some newer data. We showed that that was the case. So here are control males, and their testosterone levels. Here are atrazine-treated males, and here are the control females. So the testosterone levels in these animals were reduced to the point that their testosterone levels are no different than females. Now, this is one dose of atrazine, 1 parts per billion. This is one dose exposed to the animals for a week, and then a measurement that was done in the morning. What we've looked at now is, well, what happens over time when you're exposed to multiple doses of atrazine? And here are those data over time. And so what you'll see is that the atrazine-treated animals sort of crash and sort of never go back up compared to the controls. But the other thing that you'll see is that the higher doses actually produces intermediate effect. So if we take a snapshot and look at day 81 for these animals, so here are controls, here are the different concentrations of atrazine. Again, a non-monotonic dose response like you heard about yesterday. And what we believe is going on here is the following. At these intermediate doses, it looks like atrazine induces some of the degradative enzymes. So at these intermediate doses, it's less effective because the animal's metabolizing or degrading the atrazine faster. At the higher doses, the enzymes are still induced, but the concentration's high enough that the enzyme sort of gets overwhelmed. And at the lower doses, it appears, in some work that we're looking at now, that there's not enough atrazine here to activate those degradative enzymes. So this is a more effective dose than these higher doses. So we published an initial paper in Proceedings in the National Academy of Sciences, hermaphroditic demasculinized frogs that after exposure to the herbicide atrazine at low ecologically relevant doses. What I'm going to do here is now walk you through systematically how we sort of explored how significant or how important this problem was. Because at this point, we've only looked at one species in the laboratory, one species of amphibian. And we also couldn't get the full story in that one study. So for example, we didn't know if these hermaphrodites were males with ovaries or females with testes, which sounds like a sort of an easy question to answer, except that frogs don't have sex chromosomes. So we didn't know who was who genetically. We had some idea because, for example, in a typical study, we might get 50% females, 30% males, and 20% hermaphrodites. So we had an idea that it was the males that were being affected and not the females, at least in what we were measuring. The other thing we didn't know is we didn't know what happens when these animals become adults, because we were just looking at the little metamorphs, right? So they go from a tadpole to a little frog about this big. And we didn't know if they would remain hermaphrodites as adults, or if they would go turn into males or turn into females, which again sounds like an easy question to answer, except it takes 4 years for these guys to grow up. That means I have to tell an undergraduate as a freshman, yeah, I have a project for you, and by the time you graduate, you might have some interesting data. In fact, it was worse than that. It actually took us about 8 years to actually get this project done. The short answer is, and this resulted in another National Academy paper, the short answer is by the time we actually got the study done, by the time we actually grew the frogs up, we had a genetic marker. So we could tell who was a male and who was a female. Sure would have been useful 8 years ago to have that, but that wasn't the case. The bottom line is this guy, who sort of, to me, looks like he's smiling, is a guy, and that's his brother. So it turns out, in this population, about 10% of the exposed males literally turn into females. So we believe they start out as hermaphrodites. This guy is now a grandmother. So this is a genetic male that they lay completely functional eggs. They mate with other males, the whole thing. We probably could have published a paper at this point, because that's a pretty dramatic finding. But I wanted to know, what about the other 90%? What about the males that did not turn into females? What happened to those? Because in this population, only about 10% transform. And so what we set out to do, I have a background, my undergraduate background is in behavioral biology. And so I said, well, what kind of experiments can we do to test whether or not these males can function normally? And it required a pretty fancy and expensive apparatus. We did these experiments that I call the pool party protocol. And here's how this went. So literally, we'd put 4 females in there. We'd put 4 control males and 4 atrazine-treated males and just let them do what they do or not. And so at 6 p.m., we put the frogs in there, and we stitched them so that in the morning we can tell who's who. So there's stitches that you can see there. And then we just looked at who made it successfully and who didn't, who got the female. And everybody was a virgin. We controlled for experience and all kinds of things. So we had a limited number of time we could do these experiments, because you don't know. And so it turns out that the atrazine-treated males almost never, we only had 2 atrazine-treated males ever get the female. Okay. Turns out, because I'm an endocrinologist, you can't just observe, you have to measure something. So it turns out if you measure testosterone, on average, the controls have much higher testosterone. But then, specifically, if you looked at who made the love connection, if you look at the individual testosterone levels, there's almost a threshold. Either the females don't like these guys with low testosterone, or they just get beat up by the other males. But only 2 atrazine-treated males, the 2 that got the female, also had high enough testosterone levels above this threshold. So testosterone is kind of controlling this behavior or leading to them not being competitive. Well, then we had some animals left over. And I said, so now we know that they're not competitive, let's test if they're capable. So we did a series of experiments that I call the Motel 6 experiments. And in this case, we just got them a room, no competition. We put a male and a female here, atrazine-treated male and a female. We put blinders in so they can't see what's going on next door. And we not only tested whether or not they were capable, we tested whether or not they were competent. In other words, if they got the female, could they fertilize eggs? And again, a very complicated protocol. We left these guys together, we collected the eggs the next morning, and then 4 days later, we counted how many hatched. So I had an undergraduate, they laid about 2,000 eggs each. So yeah, there's an undergraduate, 1, 2, 3, real complicated stuff. So it turns out that if you look at controls versus these atrazine-treated animals, the atrazine-treated males have fertility rates at about 15% compared to 85% for controls. And there are a couple of reasons for that. If you walk in the room and check them out, these atrazine-treated males, most of them, just sit there and watch the females lay eggs. They don't even try. So they don't have the testosterone that motivates the behavior. If they do try, as shown here, they have very low fertility rates, and for the following reason. So here's a testis, and here's a testis from a control and atrazine-treated animal. And actually the beauty of this study is the students who actually did this morphological analysis were different students than the ones who did the behavior work, and were different students than the ones who raised the animals up. So this was all being done blindly. If you look at a blow up of here, I can show you what's going on. So this is a testicular tubule that's all full of sperm. And these guys, the testicular tubules have cellular debris, and only a few tubules actually have sperm. So they don't have enough testosterone to, 1, be capable to even try And even when they do, they don't have enough testosterone to be competent. And so we published another paper. Atrazine induces complete feminization and chemical castration in male African clawed frogs. There's 8 undergraduate co-authors on that paper. And I managed to get as many words in the title to piss off the chemical company as I could. It's also a plus for me. Now here's what's going on. And this work is unpublished. We're just about to submit this now. So I have a student. We decided we were going to repeat this experiment and look to see whether or not there was a genetic component. In other words, are some families more susceptible than others? So we have these tanks that each have 40 male frogs in them. And the student who's monitoring them will come up in the morning and say, it takes me a long time to clean the tanks. I have to pull them apart all the time. I said, what do you mean you have to pull them apart? And so here we go, there. And I knew that they're all these guys, and they're a real party. But I knew that they were all males. These are not ones that have been feminized. So what we decided to do first is, and there's a bunch of things I won't have time to tell you about, as we first decided to see, is it just big males overpowering little males because they're in these tanks of all males? And the answer is, we just went down one morning, we pulled out some guys, and we measured them, and there's who they're paired up with. So that's not the case. The ones on the bottom are always bigger. In fact, what we now know is the ones on the bottom are always the same. Turns out if you look at the testosterone levels, so after we pulled them apart, we took hormone levels, the ones on the top have higher testosterone levels typically, but more important, the ones on the bottom have estrogen levels that are typical of a female. They have an estrogen-testosterone ratio that are typical of a female. And these guys completely control the behavior. And by that I mean, if we pull these guys out, they're always the same ones, the behavior goes away. We can stick these guys in a control tank and the control males will copulate with them. We believe they've been exposed to enough atrazine that, one, they have testes, and I'll show you. They have testes that are putting out hormone profiles that look like females. And we believe that their brains have been feminized so that they have an orientation or identity that says female, even though they're male. And we've just now got the techniques working to compare male and female brain morphology, and that's the last step in publishing this paper. And by the way, there are other ways that they're feminized. So these are testes, and that's oviduct, which should not be in a male. So that's the equivalent of a man with a uterus. So it's not a hermaphrodite, it's a male, but it's got some female components, including the behavior, and we believe also the brain morphology that's associated with that behavior. We've shown, I think, that there's an effect of atrazine that causes a hormone imbalance, that generates developmental problems, that are then, because you might say, so what? That are then associated with reproductive physiological problems. Now the next step is to show, is this just something, in one species of frog, or is this something more general that we should be worried about? So the next thing that we did is we looked in the North American leopard frog, and here's the testis. This is from a paper we published in Nature. And these guys, you see all this, I usually refer to as the junk in the trunk. These are all eggs that have developed in this male's testis that are bursting through the surface of this male's testis. And I usually tell the guys, imagine a dozen chicken eggs in your testicle. That's what this animal is experiencing. So now we know that the feminizing and demasculinizing effect is not limited to this one species. What I want to do now, before I take you to the next step, is there was a lot of conversations about dose and things like that going on over the last couple of days. So I want to give you an idea of sort of the dose ranges that we're looking at in the laboratory. This is on a log scale, so this represents a 10 million-fold range, micrograms per liter, which is the same as parts per billion. For atrazine, here's the so-called NOEL, no observed effect level. 3,000 parts per billion is based on the ability of atrazine to cause a pituitary tumor in rats. So with the 1,000-fold safety factor, here's the drinking water standard for atrazine at 3 parts per billion. Okay? Here's where we start to see atrazine inhibiting growth of the larynx at 1 part per billion. Here's where we start to see atrazine, so there's different thresholds. Here's where we start to see atrazine feminizing gonads at 0.1 parts per billion. And here's where we start to see atrazine inhibiting testosterone levels, 0.01 parts per billion or micrograms per liter, well below the drinking water standard and certainly well below the so-called NOEL. Just for your reference also, here's the detection limit for atrazine and estrogen. If you use an ELISA or radioimmunoassay, right at about 0.01. So that's kind of what gives you this idea that this is a low dose. However, consider the following. Here's when estrogen starts to feminize the gonads of frogs, much lower, significantly lower, at least tenfold lower than atrazine is effective. And should I call him my colleague? Klaus Werner, who's actually a scientist for Syngenta, just published a paper showing that at the lowest dose he used, and we're getting the same thing in the laboratory, estrogen inhibits reproductive behavior at 0.0001 micrograms per liter. So like 10 million times lower than the NOEL. What this means also is, if you think about these doses, that means that you can get effects that would affect the population long before you see the morphological effects. These guys stop calling, they stop singing, females stop mating with them, they stop copulating all the way at 0.0001, something that's 10 million times lower than the NOEL for atrazine. So the animal might look fine, it looks healthy, it's not sick. You wouldn't even see morphological effects in this animal, but it clearly has a behavioral effect that will affect its ability to function and to carry a population. So the next question they ask is, do effects occur in the wild? Is this just some laboratory phenomena that, well, maybe it occurs in multiple species, but so what if it doesn't happen in the real world? This is an old slide from my old Nature paper. But again, I want to give you a perspective on levels, on concentrations. The application rate recommended for atrazine is 2.9 to 29 million parts per billion. So that means a farmer might be applying this stuff at levels that are 290 million times higher than we're using in the laboratory to produce the morphological effects, not even just the behavioral effects. This is gleaned from the literature, minimum and maximum levels in agriculture runoff, temporary pools, permanent water, precipitation. Here's our point 1 where we start to see intersexes. And so in red now are all the habitats where animals would be at risk. There's enough atrazine in rainwater. 500,000 pounds of atrazine come down into rainwater every year and can travel over 1,000 miles. My understanding is that's why the European Union, part of the reason, got rid of it, because they banned it in France and then it was still coming over into rainwater from Germany. Again, here's the drinking water standard. So we're talking about a very realistic dose. Whether or not you want to call it low dose or not, that's up to you, but we're talking about a very realistic dose. Even rainwater has up to 10 times the amount of atrazine that we're using in the laboratory. So we did a study where we collected animals from the field and we examined them. So here are kidneys, here's a testis. I'm going to show you a slice, so just imagine I'm slicing up a salami. The color's different because of the stain we used under the microscope. And here's your blow up. So this is an animal now from the field that has testicular tubules with eggs in them. We call them testicular oocytes. No sperm, just eggs. We did a study that, again, was published in Nature and in Environmental Health Perspectives, where we showed that everywhere we found hermaphrodites or feminized frogs, there was detectable atrazine. It's difficult to find a control site, in fact. And again, I would argue if these were the only data we had, it wouldn't have gotten into Nature because it's just a correlation, just like you heard yesterday. Except that we can reproduce this effect in the laboratory. We can take these animals from here and raise them in the laboratory in clean water and show that they're not naturally hermaphroditic and put them back in atrazine and make the effect return. So we've got the laboratory data combined with the field data and a little bit of mechanism, and I'll show you a little more of that later. So we've gone from showing that we have a laboratory effect. We've shown that it's not just restricted to one species. We've shown that it's not just restricted to the laboratory. We can get correlative evidence from the field, showing an association between atrazine and the same morphologies that we produce in the laboratory. What we're going to do now is we're going to take another step. A colleague of mine wrote in Ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. I've told you not just about more than one population or species of frog, I've talked to you about the animals that I just showed you are in different genera, even in different families, phylogenetically. And others have shown in other species that I haven't shown you. What I'm going to tell you now is that there are similar effects in fish, reptiles, birds, and mammals. So similar effects are occurring not just in multiple populations, but across vertebrate classes. And here's how I discovered that. I'm not going to go into it here, but some of you may know that I've suffered somewhat of personal attacks from the industry. They're convinced that I'm crazy. I'm not going to argue against that. In fact, I'm quite proud of it. But just because I'm crazy doesn't mean my science isn't bad. So what I decided to do, because they were saying, oh, it's just some crazy guy from Berkeley and frogs, don't worry about atrazine. What I decided to do is review all of the literature and show that, well, no, it's not just me and it's not just frogs, But then what I decided would be even better is what if I emailed, I'm an emailing fool. What if I emailed everybody in the world and got them to co-author a paper with me? And I didn't get to update this now. There's 62 of us have just written a big review paper from 17 countries, from every continent except Antarctica. You try editing a paper with 17 different languages. Anyway, it's been a nightmare. 62 of us now. We published a shorter version because this has been going on for a year of me trying to get this together. We published a shorter version just in the last year or so. And here's what we showed. And I didn't even know this. As much as I think I know about atrazine, I wasn't even aware. So here's my frogs, right? Sperm in the testes, empty testis if you're exposed to atrazine. This is fish from Belgium. Look at this one. These are reptiles. This is a caiman. These guys study in Argentina. Almost exact same effect. You expose these reptiles to atrazine, no sperm in the testis. This is rats. This work was done in Croatia and in Nigeria. Here's a testicular tubule full of sperm. You give the rat atrazine, there's no sperm. And what I would have said, and this is the figure we published in the paper, and what I would have said 2 weeks ago is that it seems like birds aren't affected. Then I got an email and a recently reviewed peer-reviewed paper from a scientist in Pakistan. Here's quail, sperm in the testis. You give it atrazine, the sperm's gone. So across vertebrate classes, somehow 62 scientists from 17 countries have all screwed up and produced this artifact because atrazine is safe. We're working on the bigger paper now. The other thing that's also really just coincidental because we're all screwing up is whether or not atrazine reduces sperm because it reduces testosterone. Well, it turns out this is salmon. This is a guy in England. Here's my frogs again, and here's rats. So now again, we have this coincidental effect that's occurring across vertebrate classes when animals are exposed to atrazine. Now, this is my colleague Shauna Swan, who is also one of the co-authors on the big paper. And what Shauna Swan showed a few years back, that if you look at the fertility of men, there's a correlation between low fertility and atrazine contamination in the urine. These guys have low sperm count and trouble getting their wives pregnant and low semen quality. Now again, that's just a correlation, right? Except that if you give a fish or frog, an amphibian, or reptile, or bird, or rat atrazine, testosterone levels go down, their sperm count goes down, and somehow there's just this coincidence that men who have atrazine in their urine, which by the way, 0.1 parts per billion in their urine, is associated with low fertility and low sperm count. Now, what I like to do here is I squash the data down because this is a colleague in California who's shown that here are atrazine levels in the urine of field workers. Now let me squash that down again because here are atrazine levels in men who apply atrazine. 2,400 parts per billion. Men who apply atrazine have 24,000 times the level of atrazine in their urine that my colleague's shown is associated with low fertility and low sperm count. Men who apply atrazine have 24,000 times the atrazine in their urine that we know is associated, that induces demasculinization and feminization in our frogs. Or, as I usually tell my audiences, one of these guys could pee in a bucket. I could dilute it 24,000 times and use the atrazine in their urine to chemically castrate and feminize 24,000 buckets of 30 tadpoles each. This gets at something that I'll talk a little bit about, the environmental racist or environmental justice issue, because 90% of these guys in California are Mexican American. And despite what was said yesterday about chlorpyrifos, it was originally developed from nerve gas, and these guys are still using this in agriculture and still being exposed to it. And in many communities, as many of you know, life expectancies of 50 in some cases. So does atrazine turn on aromatase and increase estrogen, like we've shown in frogs? In frogs, of course, it causes vitellogenesis, yolking, and oogenesis, egg production, in males. Well, it turns out, so here, I like this one because it looks like I love the atrazine. So here's my frogs. And again, this is from the paper we just published. This is fish, Don Tillett. It's with the U.S. Geological Survey. So fish exposed to atrazine develop testiculocytes, and this is from a group in Canada showing eggs developing in the testes of turtles. So in fish, amphibians, and reptiles, all ectotherms, atrazine producing, again, the same feminizing effect that we published on 10 years ago. In mammals, the gonads are kind of protected. Males aren't going to grow eggs in their testes. Males' testes aren't going to turn into ovaries. Even hormones can't do that in mammals. But we'd worry about mammary tumors and prostate cancer in mammals. With regards to prostate cancer, a study published in 2002 showed that in Syngenta's factory, which is in a community that's 80% African American, there's an 8.4-fold increase in prostate cancer. And these data are looking at men who work in the factory bagging atrazine and inhaling it compared to men who work in the factory but didn't work in the room where they were bagging the atrazine. Every one of these men was 50 years old. So not only was it a higher prostate cancer rate, but it was occurring typically at an earlier age than you might expect. It's funny, when I first started publishing and talking about this, their lawyer likes to write letters to my dean for some reason. I told him my daddy lives in South Carolina, but for some reason he thinks my dean can tell me what to do. And in one of their letters, he said, we don't actually put our factory in that community because black people live there. We put our factory in that community because the land is cheap and that happens to be where black people like to live. And I thought, really? You think that's better than what I said? So among many studies, there's at least one in Kentucky that shows that with a p-value less than 0.0001, women who live in an area where their groundwater, drinking waters, well water is contaminated with atrazine are more likely to develop breast cancer as compared to women who live in the same community but don't drink the well water. So again, this isn't my study, this is somebody else published it, and it's just correlation, except that when you look at rats, which I guess serve as a proxy for us, at least that's what people say when they're trying to get things on the market, testosterone goes down with atrazine, as I told you, and this work was actually done in an EPA laboratory, and estrogen levels sorry, I apologize. This was done in Syngenta's laboratory, and estrogen levels go up. At the same time, if you look at rats exposed to atrazine, there's an increase in the incidence of mammary tumors. Although the company claims there's not an increase in the incidence, it just changed the age of onset, and if it controls it live long enough, they get breast cancer too. That's like saying you're going to die anyway, we're just going to help you do it a little earlier, but different perspective, I guess. In humans, and we have some work that we've published, but I'm going to show you the original study published in 2001. If you take a human cell, in this case it's an adrenal carcinoma cell, it doesn't make aromatase or estrogen, but if you give it atrazine, it starts making aromatase and producing estrogen. So again, the same thing we've shown in fish, same thing we've shown in frogs, same thing we've shown in reptiles, birds, rats. Now human cell lines show the same response at levels that are in the low part per billion range in cell culture. So the human studies are just correlation, but it sure does seem to be falling in place with all the experimental and wildlife data. In my laboratory, we've published 2 papers looking at effects of atrazine on aromatase in human cells, but more recently, and we still haven't published this yet, for reasons I can get into if you want, but more recently we've shown that if you take human breast cancer cells and expose them to atrazine, the aromatase gene is upregulated. Now, here's what that means to me. Breast cancer is estrogen dependent typically, which one shouldn't make sense, right? Because you're getting a breast cancer mostly after menopause when your estrogen levels in your blood are lower than they've ever been in your life. Turns out though, that aromatase is very important in breast cancer because it makes estrogen locally, which can bind the estrogen receptor and cause your damaged cells to turn into a tumor and eventually spread. Turns out that the local expression of aromatase is so important that the #1 treatment for breast cancer right now is, guess what? A chemical, a pharmaceutical that knocks out aromatase and decreases estrogen so that your damaged cells don't grow and turn into a cancer or a tumor. How much sense does that make when the #1 contaminant of drinking water, groundwater, rainwater does exactly the opposite? When we're using 80 million pounds a year of a chemical that turns on aromatase in every vertebrate model that's been examined that increases and that is associated with breast cancer in humans and induces or promotes breast cancer in exposed rats. Here's where I really got in trouble because Novartis Oncology offers treatments for cancers. So the same company that gave us 80 million pounds of atrazine up to 2000, in the same year that they discovered atrazine turned on aromatase in human cell lines, started selling an aromatase blocker to treat breast cancer. So if you're living in the Midwest, taking letrozole to decrease your aromatase because you've experienced breast cancer, how is that impacted by the atrazine in your drinking water that's doing exactly the opposite? Oh, they wrote a lot of letters to my dean. I published a paper called The One-Stop Shop, Chemical Causes and Cures for Breast Cancer. and some people with law degrees were not happy. Here's the kind of thing that I'm trying to get done now. These are the top 13 cancers that you're going to get in the US. In red now, the ones that you're more likely to get if you're African American. if you're black. The mortality rate relative to Caucasians or whites are higher for all 13 of these cancers, and I can show you similar data for Mexican Americans, for Hispanics or Latinos. Here are the kinds of questions that I want to ask. Is there a real biological difference? And this is corrected for socioeconomic and access to healthcare and all that. Is there a real biological difference? Or is it because if you're black or if you're Hispanic, you're more likely to live in and work in areas where you're exposed to crap that we know causes these diseases? And I would throw diabetes and obesity in there as well. You can't even answer the question now because there are no cell lines available from the people who are most affected. There are 0 black cell lines available to study breast cancer. And the same goes true for Hispanics. In other words, when you hear these data that you're more likely to get breast cancer if you have a female relative that has breast cancer, is that genetic? Or is it because you and your female relative have been exposed to the same crap generation after, or maybe just exposed once, and then affected by the kinds of cross-generational, transgenerational things that Bruce talked about? Again, you can't even answer the questions now. So one of the things that's happened that's been exciting for me is, because I'm just a little boy who likes frogs, is studying this aquatic organism, I believe, has told me quite a bit about this aquatic organism. And much you've already heard sort of the analogy here. This individual, this animal, depends on the same hormones, literally estrogen, androgen, thyroid hormone, that this animal depends on. And in the same way that my tadpoles end up in an aquarium or a pond, trapped with all of these chemicals that it can't get out of, in much the same way, this organism is trapped in the same way. I didn't have time to tell you about it, but atrazine, because most people care about humans and rats are a proxy for us, atrazine, in addition to promoting prostate cancer and mammary cancer in rats, other studies have shown that it causes immune failure, which we've also published on in our frogs and in fish. Atrazine causes neural damage when the pups are exposed in utero. What I think is more concerning is that atrazine causes abortion because of the hormone imbalance it creates in 4 strains of rats. And this data was produced in an EPA laboratory. Of those rats that don't abort, atrazine causes prostate disease when the pups are exposed, the male pups, in utero. So these guys are born with the prostate of an old man. This was a second EPA laboratory that published these data. A third EPA laboratory, and although the author now is no longer at EPA and is actually co-authoring with me, showed that atrazine causes impaired mammary development. So the breasts don't develop properly when the daughters are exposed in utero. And then when those daughters grow up, their pups experience retarded growth and development because this generation doesn't have the mammary tissue to support that developing pup. So this rat is affected by atrazine that its grandmother was exposed to. This rat never saw atrazine. And again, that's a third EPA laboratory has published that. That's not my work. That moves me in some real ways when I think about my little girl, when I think about the fact that we may be using chemicals today, we are using chemicals today that will potentially impact our grandchildren in much the same way that we're impacted by chemicals that our grandparents used. Not things that I thought I would be thinking about because I just wanted to study frogs. Several papers have come out showing that atrazine causes or is associated with disruption of menstrual cycles and other types of birth defects in humans. I'm just going to show you one. And let me just read to you the conclusion that they published. And again, this is somebody from the Center for Disease Control published this paper. Maternal exposure to surface water atrazine is associated with fetal gastroschisis, I can't even pronounce it, particularly in spring conception. And this is just one of many papers now that are coming out showing associations between birth defects, similar to what we see in rat models. So if you're curious, this is the condition. A former student of mine now, who's an MD, has now picked up on this, is now starting to work on this issue. And so somebody asked a question yesterday, what can we do? I'm going to close with a few things. One, I helped Keith Ellison write a bill to ban all triazines, and that bill's been reintroduced this year. You can write to Keith. You can write in support of that bill. It's just been reintroduced in the last 2 weeks. There are a number of other organizations. A Pesticide Action Network is represented here that are working on these issues. A group of young students in Illinois started a Facebook page. I actually get a lot of my news about atrazine actually now through this particular page. And there are a number of petitions that I didn't write, but I've been involved in helping the people who write them understand some of the science. And you can get involved in this way as well. And then lastly, my shameless plug, on May 2nd, opening in theaters all across the US, last call at the Oasis. My story is featured in it along with Erin Brockovich, and if you can let people know, it'd actually do a lot for the film. It's the same group that did Inconvenient Truth and a number of other important films. Thank you. Oh.
All inspiring as always. We have time for a few questions before coffee. Yes, sir.
How do you filter your water to get it out?
So the question is how to filter your water and get it out. Let me first tell you why I don't like the question. Because you can imagine I get it all the time. First one is, if 10 years ago I had made a deal with Brita, boy, I wouldn't have to be scrambling to fund my lab right now. If you look on Brita Filter, it even says it removes atrazine. And we've actually done experiments where we've removed it and exposed frogs using the filter. Now here's why, the real reasons why I don't like the question. It doesn't solve the environmental problem. It also doesn't solve the problem for the people who are most affected. Those high levels in factory workers and field workers are because of inhalation and dermal exposure. So even if they had access to the information, even if they had access to filtered water, those high levels don't come from drinking water and those people who are the most affected and the most exposed. The other reason is it puts the burden on us. And it might seem like, oh, I'll buy a filter, it doesn't cost that much. But there's a class action lawsuit in 6 states where water providers are suing Syngenta to have the atrazine removed at the source. And the idea is it would be so expensive for Syngenta that they wouldn't do it. So it might seem like a small cost to us, but if we all had to do it, it's a huge cost that really we shouldn't be, in my opinion, we shouldn't be responsible for.
I'd rather live than wait.
Yeah. But yeah, for us personally, it's a solution. It's the same as buying organic food, right? But it doesn't solve the problem for many other people.
Professor Hayes.
Yes.
Fascinating stuff. I was hoping you might speak. Obviously, the focus of your research has been on atrazine, but I was hoping you might speak a little bit more about sort of if tomorrow atrazine was banned, this sort of extrapolating from atrazine for some of the other chemicals that are used, the same sort of problems. You mentioned the trizines, et cetera, but I was hoping you might speak a little bit about the broader, I mean, obviously atrazine is a massive problem in itself, but the broader implications of it.
I mean, one, so to follow up on your comment, if it was banned today, it was banned in France 10 years ago, and the levels in the aquifers have not changed. Now, that's not true in surface waters, right? I mean, it breaks down, but in the aquifers, and then there's probably a lot of factors that contribute to the half-life and things like that. But the last I saw in France, they were at least at the 10-year mark, and it was still there. So it's pretty tough to get rid of. It doesn't stick in fat the way DDT and some of these other compounds do, but it has a pretty long half-life once it's in the groundwater. We've looked at a number of other chemicals. I didn't have time to talk about those today. We've looked at the effect of chemical mixtures, of pesticide mixtures, and we've looked at effects on immune function. I have a graduate student who just finished and has a number of papers coming up that look at immune function. Once you get to the mixtures, it's very difficult. The mixtures actually produce, the ones that we've looked at anyway, produce such adverse effects that not being able to reproduce is probably the least of your problems. The immune system failure effects, the increased disease rates that we see, in some cases, actual deformities and greatly reduced growth as a larvae and as an adult, have big impacts on amphibians. And I know that there are people starting to look at mixtures in other animals and rats and things like that, and it's equally complicated. Even just figuring out what mixtures to look at is complicated, right? We did it by couple ways. One is we go to a cornfield and just look at what was applied and then make that mixture up in the lab. Another one is we just go down and go, what are the top 10 chemicals used, or what are the top 10 contaminants used? And one of the other things that we've done is we just go upstream and downstream in rivers and actually use the water. But then when you get effects, trying to figure out what's actually in it is, quite difficult.
What does it take to measure to that 1,000th the level that it's allowed to occur in our drinking water?
So the level that we can detect now reliably by what's called a radioimmunoassay or an enzyme-linked immunosorbent assay is, that's the 0.01 micrograms per liter. But gas chromatography mass spec, which people use, typically can't measure down to that level. And measuring below that level, there's no technology that I know that you could do that. I mean, maybe there's a chemist here that knows better than I do, but if there is, they've kept it a secret because I've never seen anything measured below that level.
You had an opportunity to talk about the work you've done in the San Joaquin River.
Yep.
From the headwaters where the water was cleaner and all the way down.
Yes.
I found that just an incredibly compelling body of work and findings. Can you just briefly describe what you did in that, and have you been able to keep that work going?
Yeah, and one of the things that I try to do, sort of as a philosophy, is if it's my graduate students' work, I kind of let that be their program, so that they're, so that work is mostly my graduate students. And what they've done is we have 3 rivers in California: the Salinas River, the San Joaquin River, and the Sacramento. And what we've been able to do is to go upstream of the river and look at animals, and downstream, so that each river is its own control. And what we've shown are some pretty dramatic, like inhibition of growth, and we have other ways to look at immune function now. Greatly reduced immune function, greatly reduced male sex characteristics. And then an interesting twist on the Salinas River, turns out downstream, the males have higher testosterone. And it turns out that pool party assay I just showed you, if you raise Xenopus in water downstream, they actually outcompete the controls. So we're wondering if there's some anabolic androgen that's been used in cattle or something, who knows what it is, but it actually seems to be masculinizing somehow. But again, how do you figure out what's actually in the water? And also keep in mind, especially in California, what's coming downstream is going to change depending on the time of year as well. So you could be getting estrogenic chemicals coming down one part of the year, neurotoxins one part of the year, and whatever these things that seem to really make the frogs macho coming down a different time. So that's 2 of my graduate students have completed that work. One looked strictly at the reproductive aspects, and one looked strictly at the immune function, but used all the same sites to do it. And that work, one paper submitted now, that work should all be coming out pretty soon.
So at the beginning of the week, the scientific journal USA Today published a front page story about the fact that teenage pregnancy rates in the U.S. are at a historic low, never ever recorded in the past. None of the sorts of issues that you raised in your talk about potential effects on human fertility were addressed in this publication. Yeah. I'm wondering, and particularly given what we know that Shauna Swan found in 2003, it would seem that there might be some relevance. I'm wondering if you would be willing to reflect upon that.
I would agree. I mean, of course, you know about the studies going way back, looking at sperm count in Europe and as well as in North America. So I wouldn't be surprised if there's a component of decreasing male fertility in teenagers. I actually hadn't thought about it till you just said that, though.
Well, let's think about it more over coffee. Let's try to invite guests.